So I hope everybody had a productive lunch discussion and got a chance to look at some of the posters. We're going to kick off the afternoon session, and we're going to have a couple of clinical talks and end with an exciting panel discussion. So to begin our clinical talks, we're going to start with Dr. Debbie Persaud, who is a professor of pediatrics here at the Johns Hopkins University School of Medicine. As many of you know, she's internationally recognized for her research on barriers to and strategies for a cure, HIV-1 cure in the pediatric population. She's also the scientific chair of the International Maternal Pediatric Adolescents AIDS Clinical Trial Group, or IMPACT. So today I'm very excited to hear her speak to us on an overview of clinical trials towards HIV remission and cure for children and adults. Please join me in welcoming Dr. Persaud.
Well, good afternoon, everyone. Thanks to Ashwin and Christine for inviting me to give the update, not just on pediatrics, but children and adults. And in the spirit of the title of this session, in terms of is the cure around the corner or miles away, I thought I would add a caveat, hope or hype, or it could be hype or hope, depending on if you're a pessimist or an optimist. But I do want to start by acknowledging Bob Silcano, who, as you've seen, has generated multiple clones of himself in this room from our presentation. But Bob really pioneered the work of HIV latency, its clinical relevance in people and including in children, and continues to push the envelope to really help guide the clinical trials development pathway and biomarker measurements. So Bob, thank you for all you've done to mentor us. So I know you've heard this a lot, definitions, but some of you may not have attended the morning session. I think it's just in the spirit of really for you to make your own decision whether we're on a path in terms of hope or hype. We'll start with the basic definitions in terms of the latent HIV reservoir that is the major barrier to cure. Janice Clements described the macrophage monocyte population, but the larger reservoir to date is in resting memory CD4 T cells. These cells harbor transcriptionally silent, integrated, replication-competent provirus that have this ongoing potential to refuel HIV replication if antiretroviral treatment is stopped. So with respect to HIV cure, this would be the state. This is what we're aiming for in the next 20 years, perhaps, where all cells harboring replication-competent proviruses that are inducible are eliminated from the infected person. And then the state of HIV remission, as Joel Blankson described, is a state where the reservoir is really reduced sufficiently to allow viral loads to remain undetectable even when antiretroviral treatment is stopped. And to really regroup and reframe the current goal of HIV therapeutics, which is really good, has revolutionized the lives of HIV-infected persons. The current goal of antiretroviral treatment is really to maintain the steady state of virologic suppression. Currently, the clinical assays detect down to less than 20 copies per ml. So this is our gold standard. This is our metric for antiretroviral treatment. That's combination antiretroviral drugs. We know from the seminal work done by multiple groups, including the Silcano groups, that current antiretroviral treatment does not eliminate the latent HIV reservoir. These cells undergo stochastic reactivation continuously to produce virus, but because of the potency and efficacy of antiretroviral treatment, combination drugs, these viruses that are produced are not capable of generating new cycles of infection, and hence patients remain suppressed on antiretroviral treatment. So at least we have a trajectory in terms of what to measure with current antiretroviral treatment regimens. And really the armamentarium is quite vast, starting if you look back 1987, where zidovudine was the only antiretroviral drug available for treatment. Now there are about 27 antiretroviral drugs that target 6 different steps in the virus life cycle that are approved, FDA approved for treatment of HIV infection. And these drugs have been now combined into single pill once-a-day regimens that really has revolutionized the state of HIV treatment for adults largely. We're hoping to be able to get fix those combinations for children because they can benefit from this. And we know, and Joel showed the slide on the left, that I think what is really remarkable, unlike the SIV model that Guido presented today, is really, if you look on the far right in 2015, what's really remarkable is if patients are treated continuously for up to 10 years, we see the same pattern of predictable rebound within 2 weeks of stopping antiretroviral treatment. There are some hints from a recent study from Dr. Fauci's group in a therapeutic vaccine studies that using more contemporary potent regimens that this kinetics of rebound may be somewhat delayed in patients treated during acute infection. Nevertheless, most patients rebound when antiretroviral treatment is stopped. So where we really have to think about an HIV cure agenda is for adults in 2017. The study that was published really highlighted that life expectancy for HIV-infected adults nears normal limits with treatment. And so what you see on this graph in red and blue are men and women. Those individuals starting treatment in 2008 to 2010, HIV-infected adults can live well beyond age 70 and 75. So that's where these numbers come from in terms of near normalcy with antiretroviral treatment. So when we think about giving patients different classes of drugs to purge reservoirs and the potential side effects, in addition to analytic treatment interruption, as you heard today, which is the mainstay of assessing HIV remission and cure, we do need to contextualize the state of standard antiretroviral treatment for adults. Now, for perinatally infected youth, as Lynda said, there are 1.8 million children under 15 living with HIV. We still have about 160,000 new infections annually, despite dramatic progress in mother-to-child transmission. This is really a dismal picture with perinatal HIV infection. The mean age of death is actually 23 years. And these are actually kids that were born in the early 90s. So it's possible that with our current early antiretroviral treatment strategies that we could lengthen the lifespan for these children. But an astounding number, which is really, I think, for certainly pediatric populations, it's really critical to think about a cure agenda, is that the standardized mortality ratio compared with the general US population is about 32-fold higher. So it's imperative that we think about beyond art for this population. And so the whole goal now is to shift the HIV treatment paradigm from the present state, which is lifelong art for virologic suppression, to the future HIV remission, as I discussed earlier, which would be defined as years off art without detectable viremia, and ultimately a lifetime of no art in the setting of HIV cure. So we do have sparse scientific evidence that this can be accomplished in patients. And so on the far right is the Berlin patient, Timothy Brown, and on the left, the Mississippi baby. So Timothy Brown is the patient who's been cured of HIV, as far as we know, has been 10 years in remission with no detectable circulating HIV-infected cells, but does take, as he would admit, PrEP for prevention of infection in himself. And then the Mississippi baby, who we described in 2013, who is the best example of what we would aim for in terms of HIV remission in our clinical trials. And I'll go through quickly in detail some of these cases. And not to bore you with the details, but I think it's important to point out that when these cases, in the case of Timothy Brown, this was an intentional treatment strategy for this patient where his oncologist specifically looked for CCR5 delta 32 cells, homozygous cells, to prevent him from reestablishing infection if virus was still persistent. And now we know in 2018 that he has no detectable circulating virus. And so what you see here is a curative regimen that has been used, but I think this is our case that HIV can be a curable disease in the setting of transplantation and all of the other aspects of chemotherapy that was included here. Now, I have to spend some time on the case of the Mississippi baby because I think it's underappreciated that this was really a case of serendipity. And had this case presented to us a year before, 2012, we probably would not be thinking about remission and cure. But the field was very focused in 2012 on developing new strategies towards remission and cure. And so we already in the pediatric cure community had established a definition as what we thought HIV remission and cure would look like in pediatrics. And our definition was simple. It would be a child who had received an intervention, went off treatment, and then did not experience viremic relapse, at the same time would maintain normal CD4 levels and have normal response to routine childhood vaccinations. That was our basic definition. So when we were presented with this case of an infant with known HIV infection who had presented 5 months off antiretroviral treatment and had no detectable signs of infection by antibody testing, DNA testing, latent reservoir sampling, we decided to not restart antiretroviral treatment. And that was really the basic question: should this child be restarted in antiretroviral treatment? And this child went for 27 months without experiencing viremic rebound. So really is the representation of what HIV remission should look like. And so clearly this is a child who had established HIV infection as illustrated by the biphasic decay in plasma viral load levels with antiretroviral treatment, and then creating this picture or snapshot of what HIV remission would look like. And so we continued to follow her with monthly viral load measurements, and then unfortunately 27 months later presented with no indicator signs with rebound viremia, just detected on routine viral load monitoring. And so this is the state of HIV remission and cure research. There's no identifiable to date surrogate biomarker that's predictive of HIV remission and cure or predictive of time to rebound. And so essentially patients will be taken off therapy and as part of our clinical trials and followed very frequently with weekly viral load measurements, just waiting to see how long it takes the virus to come back. And in the setting of the Mississippi baby, the reemergent virus was really a clonal population of virus. And so the overarching, so this case actually highlighted that if you can reduce the reservoir sufficiently, the intervention here was very early treatment in the first 48 hours of life, really at 30 hours of life, with continuous therapy somewhere between 12 and 18 months before really going off treatment and no rebound for 27 months. So this idea of reducing the size of the latent reservoir and its implications for long-term control is illustrated on this slide. And Alison Hill with Bob Stolkanas provided beautiful mathematical models to aid the field, the clinical trials field, in terms of what should be our targets. And currently, the rough target is about a 3-log reduction in the size of the latent reservoir. And we have representative patients here showing a Berlin patient who perhaps experienced 6 or greater log reduction in the latent reservoir that's afforded a cure. And then the Mississippi baby, based on our IUPM measurements, we estimated we had about a 3-log or more drop. And then there are two patients from Boston who were transplanted who also had decreased reservoir size that rebounded but at 12 weeks and 32 weeks, not the 14-2-week period following ART interruption. So what you see on this slide are the current therapeutic strategies towards HIV remission and cure. And the one end of the spectrum is very early and early antiretroviral treatment. You heard about latency reversal today. Immunotherapeutics, which Katie will talk about. I think this is the most promising part of the HIV cure agenda, with broadly neutralizing antibodies and therapeutic vaccines, and gene and cell-based therapy, so disrupting the target cells so that these cells are not susceptible to HIV infection. Now, there's also modalities that are being tested, target inflammation. Patients who are HIV infected, despite effective control of virus replication, maintain this state of heightened inflammation and immune activation. There's immune exhaustion. Joel touched upon the real barrier here to cure is really cellular proliferation, and Guido also touched upon the homeostatic proliferation or proliferative capacity of these cells. That's really a major barrier to clearance. And then immune replacement via transplantation for patients who have malignancies. So I will say we're really in the case reporting stages of HIV remission and cure therapeutics. What you see on the left-hand side are the patients to date who have achieved remission with very early antiretroviral treatment, with the first example being a perinatally infected the Mississippi baby. The Visconti cohort that Joel Blankson mentioned today, 14 post-treatment controllers treated during primary infection in the French cohort. There's a French adolescent who is perinatally infected, treated for 6 years, went off treatment, and is now off treatment for 14 years and remains in remission. There's a South African SHARE trials participant who is about 9 years old, was treated for 40 weeks and has remained off antiretroviral treatment. And then this was recapitulated, this concept of very early treatment, reducing reservoirs, by the San Francisco PrEP patient who was treated within days, I think, was on PrEP, got infected, had a peak viral load of 230-something copies, remained on treatment, but went off treatment and rebounded 8 months after stopping treatment. But together, these cases really confirm the notion that we need to reduce replication-competent reservoirs sufficiently to afford some time off antiretroviral treatment. Now, what's a meaningful time frame? Is it 6 months, 8 months, or a year? That's really hard to identify, and I think we need to try to do some cost effectiveness here in terms of the viral load monitoring stuff to see when does it really become, what's a reasonable time frame for us to shoot for. So in our perinatal trials that I'll discuss later, our time frame is a 48-week off antiretroviral treatment with no rebound. On the far right is really showing immune replacement can delay viral rebound, and there's been a recent report of a Minnesota patient who rebounded after 8 months off antiretroviral treatment. And so the current approaches to reversing HIV latency is shown on this slide. These are challenging therapies. It really requires the main agent that has been studied so far, histone deacetylase inhibitors, mainly vorinostat, romidepsin, and panobinostat, and I'll show you a snapshot of some of those data. Liang showed one data slide from the latency reversing effects of these HISDAC inhibitors, but there are also TLR7 agonists, TLR9 agonists, IL-15, as you heard about earlier, that are in clinical trials. Similarly, I think this SIMAC mimetics are very interesting in that these reactivate, they're NF-kappa B agonists, and more recently, Ann Sharudi with Guido showed that in the NHP models that these are really potent at reversing or reactivating HIV. So there are many drugs that are in armamentarium that are to be added to standard antiretroviral treatment to reverse HIV latency. The goal here is to force the virus to express itself, messenger RNA protein expression to be killed either by virus-induced cytopathic effect or effector or humoral mediated processes. So no one showed this slide today, and it's always present at every meeting, but it is, what are we trying to do here? You know, you heard the mean frequency is 1 infectious units per million. That was reported by Janet Silicano in 2003 in adults, and in 2015 by Crooks and Nancy Archin's group showing the same half-life. So this is what we're trying to do. We're trying to increase the clearance rate of the latent reservoir so we can get to a point of 3 log reduction or more, a very challenging endeavor. This is the first study I did when I joined Bob's lab to really illustrate that the reservoir dynamics are very similar in children who are treated during chronic infection, meaning they didn't have access to antiretroviral treatment at that time. ART was not available to treat for treatment of kids, but then when it became available, we showed that these kids starting treatment at 8 to 9 years of age had the same stable persistence at 1 infectious units per million. I was intrigued that in the NHP model that Janice Clements reported that it's the same 1 infectious units per million. So I don't know what's about the 1 infectious unit per million, but we're seeing that across the board. Now, a ray of hope is that standard approaches to antiretroviral treatment in children now is early antiretroviral treatment. And early antiretroviral treatment is defined as starting therapy at 2 to 3 months of age. Why 2 to 3 months of age? All infants get post-exposure prophylaxis with 6 weeks of antiretroviral treatment, 4 to 6 weeks, depending where you live. And it takes about 6 weeks for there to be sufficient ramp-up of virus replication to be able to readily detect by DNA PCR HIV infection status in these infants. So it is a global standard that once you identify infection in children, irrespective of viral load, CD4 therapy is started, and we've had experience with this for the past 20 years in the United States. And what's really refreshing and very promising for this group is that actually DNA dynamics of reservoirs are very different when you treat early in kids. And what you see after 16 to 18 years of suppression, perhaps even starting at 10 years of age, is that the proportion of infected cells in the circulation, we don't have any tissue samples in these kids, drops dramatically by the 2 logs. So we're starting out with these long-term early treated kids with very low proviral reservoirs. They have no minimal HIV-specific immune responses, suggests that there's very little inducibility of the proviral genomes that persist in these kids. So I'm just going to review now some of the findings from proof of concept clinical trials of latency reversing agents, just to bear in mind that the sample size for these studies range from 6 to 17. And what you can see with the acetylase inhibitors is that uniformly using 3 different kinds, vorinostat, panobinostat, and romidepsin, you can induce HIV cell-associated RNA. But as Liang pointed out, this induction does not lead to clearance of HIV-infected cells. So this is disappointing news. We can reverse latency, but it doesn't affect the size of the reservoir. And I think as a field, we need to kind of think about at what point, and this is my own opinion, at what point do we, as we move along the pipeline of these agents, at what point do we think that these agents should not go forward if we're not seeing an effect? Now, importantly, with the romidepsin study, what they found when combined with therapeutic vaccines in a small subset of patients, 6 patients, there was a 38% reduction in the replication-competent reservoir. That has not been replicated by other groups, but I think it's intriguing that combining romidepsin with a therapeutic vaccine led to detectable reduction in the latent reservoir. So that's with respect to latency reversing agents. We've learned from these trials, small trials, that LRase alone is insufficient to clear the reservoirs, but perhaps combining with immunotherapeutics, such as therapeutic vaccines, we can affect change. And I'm not going to go into detail because I know this is the bulk of Katie's talk, but this is a very exciting field of these broadly neutralizing antibodies that are antiviral, they're long-acting, they target multiple strains, and they target multiple sites on HIV envelope. And just to kind of give you a snapshot in terms of the administration of VRC01, which is a CD4 binding antibody, in terms of its effects in chronically infected patients, it was actually not substantially effective in delaying virus rebound. You see a very similar return of virus rebound, but I'm sure Katie can identify unique patients that may glean some insights into how these antibodies are working. Similarly, it was disappointing that patients in Thailand treated during acute FEBIG1 stage infection, getting VRC01, a broadly neutralizing antibody, there was really no substantial effect on time to rebound in this study. There was 1 participant who went up to 42 weeks in this study who before rebounding, and I think there's always 1 participant in these studies that's an outlier, but together I think we can begin to think about the ones who've responded and try to learn what was different about those patients. And this is exemplified by the gene-edited cell study done by Pablo Tabas, in which autologous CD4 T cells were treated with zinc finger nucleases to render those cells CCR5 delta 32 and reinfused into patients. And after reinfusions, the patients underwent treatment interruption. The total sample size for this study, I think it's actually fascinating. These cells can be modified. They persist for up to 42 weeks. You can detect them in mucosal tissues. There was 1 of 6 patients who did not experience viremic rebound within the defined 12-week interruption period. And it turns out that patient was actually heterozygote for CCR5 delta 32. So I think each of these studies, we can learn something from the single patient that appeared to have a different or an unexpected result. Now, in terms of therapeutic vaccines, so I think the messaging here is that we really haven't seen any single strategy that has move the needle in terms of time to viremic rebound with single cases. And similarly, this is a recent study published by Dr. Fauci's group looking at patients treated during acute early infection with small reservoirs, therapeutic immunizations, a vaccine placebo group with rebound viremia. Now, in this study, again, I think we also need to take stock when we take patients off antiretroviral treatment. In this study, the patients receiving the vaccines had no identifiable immune response to the vaccine and underwent treatment interruption. So it may not be so surprising that the rebound time was not affected with early treatment. And we need more studies like this where there is a clear biomarker of effect, so immune responses in a therapeutic vaccine trial before one would take patients off treatment. So this is very great news. I think right now they're about, and these are data from Richard Jeffries, who spends hours trying to keep the treatment action group website up to date with all of the clinical trials. So any of you interested in what sorts of agents are being tested, you can find it on his site. So there are 93 intervention trials in 26 countries being conducted. Most of these are in the US and in Australia and in Thailand. But through our pediatric program, we actually have most of our cure research agendas in sub-Saharan Africa and Latin America, because that's where the burden of disease is. So what you see on this slide, this is really wearing my pediatrics hat. What you see in yellow is really the diversity of the research portfolio for HIV remission and cure in adults. Not that all of these are applicable to children. You know, we have to be very cautious. As Jeremy said, you know, we have to think about the risk benefits of moving this treatment strategy in younger populations. But we do have very pivotal studies that I think will inform the field, shown in blue. And that is very early antiretroviral treatment, and then the first studies of combining early treatment with broadly neutralizing antibodies, namely VRC01. With Lucio Gama, we've been able to now work on establishing PK data for VRC07523LS, and hopefully we'll get support for combinations of these antibodies in the pediatric setting. So just to kind of give you a snapshot, this is ACTG studies of HIV cure that's shown on this slide. It's treatment of elite controllers, use of LRAs, use of anti-inflammatory agents, and also combination and therapeutic vaccines. So the immunotherapeutic strategies are completely reasonable to start in pediatric populations once the products have been tested to be safe. And so it's not as if we don't have clinical trials. We actually have 9 clinical trials in the field for HIV cure research, heavily reliant on very early treatment. What is very early treatment? It's treatment within 48 hours of life for these infants, with addition of broadly neutralizing antibodies. And in addition, there's a UO1 looking at therapeutic vaccines in kids living in Europe, South Africa, and Thailand that's in development. So I'm going to wrap this up, but just to kind of give you a sense of these trials are very labor intensive. So this is our P1115 trial that's fully enrolled, 440 mother-infant pairs. We have 54 identifiable infants treated in the first 48 hours of life. Unfortunately, our transmission rate, in utero transmission rate, is still about 8 to 9% in high-risk women. So we have a lot of work to do with prevention, but also we have this opportunity to intervene with very early treatment. Our second trials is really an extension of P1115. So within P1115 now we've added an integrase inhibitor combined with VRC01 to do a similar analysis of treating through 2 years of age before stopping antiretroviral treatment. And then in this study, 2008 is VRC01 through 14 weeks of life. So I want to end with the hype and the hope aspects of this. The case reports, they come with a lot of hype, but they provide a lot of hope. And what you see on this slide is this ongoing report, and you heard from Joel Blankson today, patient 169, who I should add to this slide, but there are these cases of remission, sustained remission in various patients that provide hope to the HIV-infected individuals living with HIV. So what we hope to end up with is really this timeline in terms of pushing HIV-infected individuals towards a post-treatment controller state so they can maintain control without antiretroviral treatment. So the hope here is that there are really 37 million people living with HIV, 1.8 million children. I think we need to think about the context of lifelong antiretroviral treatment and the complications with that treatment approach, particularly in perinatally infected children. So I'll stop there and acknowledge the impact committee that spends a lot of time developing these protocols and implementing the protocols, and acknowledge Hannah Gay and Catherine Luzuriaga for identifying the case of the Mississippi baby, who really was the catalyst, like Timothy Brown, for the HIV cure agenda in pediatrics, and the people in my laboratory who spend tireless hours doing these assays on pediatric samples, and the funding agencies. Thank you.
So I think we have time for a question or two for Debbie, if anyone has a question.
Debbie, thanks for a really excellent talk. I wondered if there's some lessons to learn from the childhood cancer, and particularly thinking about targeted new reservoirs such as the CNS, and if there are any specific strategies that would try to attack the cells in, for example, the CNS over time, long term.
Yeah, thanks, Mary Glenn, for that question. So that's a two-part question. So I think in terms of kids with cancer, we do have an impact protocol, P1107. That's a cord blood transplantation protocol with delta 32 deleted cord blood cells. It's an opportunistic protocol that's enrolling children through adults who've received, who require transplantation for other purposes. So we hope in that study that we will be able to better characterize reservoirs in the context of transplantation with resistant cells. We have enrolled 2 adults in that study. 1 adult died at 1 year post-transplant. But we do have a single participant who actually has the same tumor as the Berlin patient. She's received cord blood with CCR5 delta 32. It's 1 year out, and we're very excited. It's doing very well, and so we'll begin the studies of HIV persistence in her and to be able to evaluate the effects. The second part has to do with the central nervous system. And it's been very difficult. I think Jeremy touched on this in terms of invasive procedures in children. But for children, it's actually very easy to justify spinal tap. Neurocognitive outcomes, as you know, is a major complication of HIV infection in children. I mean, most kids function 2 standard deviations below the norm, even with early antiretroviral treatment. So within the impact, the way we had to sort of sensitize the community to receiving spinal taps, and Anne Charuti is leading, as part of our impact, we've now started to do spinal taps in adolescents, actually kids 13 and to age 24, to begin to look at the CNS as a CNS escape and as a sanctuary site. We think as sites get comfortable with doing spinal taps, that we'll be able to move that down to the younger age group. But it's, it's the only neurotropic viral infection I know about that there's such reluctance doing spinal taps. But I think as the agenda blossoms, we'll be able to perhaps even look at lymph node fine needle biopsies to really understand the pathogenesis of rebound, because most kids rebound within 2 to 4 weeks. Thank you for that question.
Debbie, thank you for a beautiful talk and all your pioneering work on pediatric infection. You presented some very sobering data on life expectancy for perinatally acquired infection. What about children or adolescents who acquire the infection and the sort of prognosis nowadays? Yeah, so
in that study, 86% were perinatally infected, but actually 14% were not. And, it's hard to tease out the outcomes, but I think it's very important to understand, that's a population that's very difficult to have adherence on a regular basis, but they're very keen on participating in HIV research. So, the reservoirs established is actually very interesting. It's quite clonal in the first 2 years, even if they start therapy at lower CD4 counts. I think we have a lot to learn, and we need to really understand the life expectancy outcomes in those kids, because they're not as compliant with therapy and could benefit from a cure or, a broadly neutralizing antibody strategy that's every 6 months or so. But thank you for asking that question.
All right, so I'm very excited about our final speaker today, who's Dr. Katharine Bar. She's an assistant professor of medicine at the University of Pennsylvania, and there she also directs their CFAR viral and molecular core. Katie's a clinical virologist, and she led one of the major interventional trials in humans for HIV-1 cure, looking at whether administration of a monoclonal antibody could delay HIV rebound, which was published in the New England Journal in 2016. So we're very excited to hear her speak today on viral and immune dynamics of rebound from latency. Please join me in welcoming Katie.
All right, well, I apologize for the late change in title, but I'm just going to talk about broadly neutralizing antibodies in the context of HIV treatment and cure, because I think that's what you originally asked me for. Anyway, it's really wonderful to be invited here. I really appreciate it. And oh goodness. All right, I have no disclosures, so we'll go from there. All right, so to talk about broadly neutralizing antibodies, I just want to start with the concept of the antibody response. So as we're all aware, HIV envelope is actually really challenging to neutralize. So the envelope spike is covered with a glycan shield. The exposed regions of this envelope are highly variable and quickly escape from any immune responses. The epitopes that we want to target are actually recessed, conserved, or masked, or only transiently exposed. In addition, our antibody responses are hampered by the immune dysfunction of virus attacking our CD4 cells. So when we think about the autologous antibody response, we think about it as sort of being too little, too late at every stage. So for the first several months of infection, all we really have are non-neutralizing antibodies. And then within a couple months, we do see some strain-specific neutralizing antibodies, but because they're very specifically targeting these exposed regions, the virus is able to quickly escape, and these responses are not particularly effective. We do see broad neutralization arise after several years in a minority of patients, but it's not actually helpful for those individuals. However, we have been able to look at those antibodies, to clone them, and to characterize them, and that's sort of where this whole field of bNAbs as therapeutic has arisen from. So if you think of a brief history of bNAb discovery, the first antibodies were characterized in the early 1990s, and we were able to identify antibodies targeting several different epitopes. Compared to what we have right now, these were relatively modest in terms of breadth and potency, but it was proof of principle that the immune system could generate these broadly neutralizing antibodies, and it was really a big step forward. Fast forward about 15 years, and several enabling technologies allowed the field to really exploit broadly neutralizing antibodies. So high throughput neutralization assays, as well as B-cell culture and cloning techniques, allowed investigators to screen hundreds and hundreds and hundreds of patients' plasmas until we were able to identify this next generation of very broad and potent monoclonal antibodies. And so if we think about where we are right now, we have more than 7 classes of bNAbs, each targeting a distinct conserved epitope. There are at least 4 of these classes that are now moving forward in sort of relatively late stages of clinical development. And thanks to Lucio and our friends at the VRC and other partners, we're able to start testing these in clinical trials and looking at how they may be able to act as almost drug agents. So these bNAbs, of course, have unique biology that makes them able to access these sort of conserved and recessed epitopes, including extensive somatic hypermutation, long CDRH3 arms, and auto-reactivity. So these are the exact properties that make them very challenging to arise during natural infection and challenging to induce via vaccination. But the whole concept behind exogenous administration of these antibodies is that you could circumvent the immune system and thereby allow a highly effective mode of either prevention, treatment, or cure of HIV infection. So where are we right now? We currently have about, like I said, 4 classes that are at or near clinical development. And so their CD4 binding site antibody is one of our most developed. We have actually 3 or 4, at least 4 antibodies that are currently in development. The V3 glycan, we have 2 antibodies in clinical development. The membrane proximal external region down here has a single antibody in development. And then we have 2 V2 or apex glycans. In addition to these natural antibodies, however, we have several engineered antibodies or antibody-like molecules that are coming in the near future that are pretty exciting. So we have both bispecific and trispecific antibodies where they're able to combine targeting of multiple moieties into a single antibody epitope. And then there's immunoadhesin type molecules that work on an antibody-like framework to be able to bind, for instance, with the example of eCD4IG, both the CD4 binding site and the co-receptor region to provide a lot of breadth and potency. Now, these all offer a lot of promise. They each has a range in its breadth and its potency, and they're all in various stages of development. So it's going to be an iterative process as we move through the next few years to see how these all pan out. But I think one of the questions that a lot of people ask is, well, what is the big deal about BNAbs? If this is just a long-acting antiretroviral, how is this really going to advance the field both for treatment, but specifically in the area of cure? So, our standard of care, both for prevention and treatment right now, is small molecule antiretrovirals. And as has been stated multiple times today, we're at a pretty advanced state. We have high efficacy agents that are potent with a high barrier of resistance and relatively low toxicity. And for both prevention and treatment, we have once daily single tablet formulations available. So what are the basic properties of BNAbs that may hold potential advantages over small molecule antiretrovirals? Well, the first 2 are sort of logistical. And the first is that long-acting possibility of monoclonal antibodies. So at baseline, most monoclonal antibodies have a half-life of about 2 weeks. However, mutations to the antibody, for instance, this LS mutation that most of our BNAbs now have in the FC region, extend that half-life quite a bit. There's also the possibility with development of vector delivery techniques such as AAV, so vectrovirals, or DNA or mRNA sort of genetic technologies. We could give these antibodies over the course of years or many, many months, and therefore they would be quite long-acting. Monoclonal antibodies, in addition, are actually quite well tolerated. So when we think about using these across the landscape of clinical medicine, monoclonal antibody therapy is more specifically targeted and tends to have fewer side effects. But these next 2 properties are what we really think about in terms of enabling BNAbs to be a component of a cure strategy. And that's that in addition to sopping up free virus and preventing infection of vulnerable cells, monoclonal antibodies can also recognize virus expressed on HIV-infected cells and therefore can clear infected cells in addition to free virus. And the second, or the last component here, is that these antibodies are a part of the human immune system. So they can actually engage with the host immune system and hopefully boost immune responses in a positive way. So if we think specifically about those mechanisms. So again, neutralization is the primary characteristic, and that's what these antibodies have specifically been selected for. So they very nicely bind to free virus and prevent infection of vulnerable cells. But we also see that they can bind to envelope if expressed on infected cells and engage through effector functions various immune cells to either lead to ADCC via NK cells or phagocytosis through macrophages or just any type of ADCVI. So finally, this concept of immune modulation or a vaccinal effect. And this is the concept that if you have large quantities of this virus and antibody bound together in immune complexes, this will engage with APCs or dendritic cells and sort of enhance immune responses both on the T cell side of things through cytotoxic or other T cell functions, and also with humoral immunity. So as we think about BNAbs as a component of cure strategies, this concept that they could be a component of the kick and kill strategy is what I want to talk about a little bit today. So they could provide, in terms of this kill part, BNAbs can sort of replace antiretrovirals and maintain suppression of viremia while also enhancing the ability to kill HIV-infected cells that are a component of this reservoir. part and parcel of this is we would still need a highly effective and safe and efficient kick mechanism, and that's still to be identified, as has been discussed in several studies today. So how do we go about demonstrating whether BNAbs can have a role in cure? So the rest of the talk, I'm going to try to talk about non-human primate and then clinical trials that have been able to demonstrate whether this is possible. So the goals in these trials are to demonstrate first that these antibodies can sort of replace antiretrovirals and that they can maintain durable suppression for a long period of time with both safety and with hopefully ease of administration and less frequent dosing. Second of all, the goal is to see if these antibodies can enhance HIV-infected cell killing and immune responses to either reduce or suppress that latent reservoir from rebounding. So if we think about animal models, I'm just going to talk about a couple non-human primate studies. And these non-human primate studies have definitely demonstrated an antiviral effect and the ability to suppress virus for short periods of time. And there's very early evidence that is controversial and needs to be really carefully considered, but evidence that these antibodies in certain contexts can lead to either reservoir suppression and or clearance. And finally, human clinical trials, of which I've been lucky enough to participate, are kind of in their infancy, the AZT age, as was mentioned earlier. But we have been able to show the effects of monotherapy and dual therapy in human clinical trials. All right, so looking at non-human primate studies. The first of these trials looking at administration of these next generation BNAbs began in about 2013. And there were basically 2 papers published in the same issue of Nature that address this. The first that I'm mentioning here is by Dan Baruch's group. Basically what they did is they took HIV-infected macaques that were viremic, and you can sort of see their viral loads coming in. They gave them a dose of PGT-121, which is a B3 targeting broadly neutralizing antibody. They gave it alone or in combination with other antibodies. And what they saw was a significant antiviral effect. So you saw the viral load come down by 1 to 2 logs. In the animals with the lower viral loads, you saw some suppression of virus below limit of detection for a period of time. And in the higher viral loads, you saw the virus quickly rebounded. Now, in this sort of unique SHIV model, they were also able to show some interesting enhancements in T cell and B cell function, but this was also in the context of suppressed viremia for a period of time. But this was hopeful that the antiviral effect and potentially immune modulation might be going on. Also reported in the same issue was a similar study conducted by Mal Martin and the Nussensweib group looking at 2 antibodies, 3BNC, 117, which targets the CD4 binding site, and 101074, which targets the V3 region. And they also gave this to chronically SHIV-infected rhesus macaques. And what you can see here, the green and the blue are the levels of antibody, the red is the level of virus load, and they saw that virus was suppressed for a period of time. And once these antibody levels waned, the virus rebounded. And so they saw a very nice ability to suppress virus for a short period of time. And when these viruses did rebound back through this antibody pressure, there was the detection of escape mutants or virus resistance to these antibodies in some of these animals. So this is sort of the first demonstration that these antibodies could suppress viremia. But it's only been in the past year or 2 that we've seen these other sort of properties of antibodies tested in the non-human primate model. So last year there was a paper published by Mal Martin's group and again with the Nussensweib group looking at the effect of these 2 antibodies, again 3BNC 117, in the context of very early administration with antiretrovirals. So what they did was, again, they took these SHIV-infected rhesus macaques, and very early in infection they started antiretrovirals plus or minus these antibodies. And what you can see in the gray line here is untreated viremia. So in theory, this is the normal viral kinetics. If the animals were given just ART, they were suppressed in this red line for a period of time and then rebounded. But if they were given ART plus these antibodies at this very early time point, what they saw was, at least in a subset of these animals, durable suppression of this virus. So the virus did not rebound. They went ahead and they took these quote, controller monkeys, and they gave CD8 depletion, or they depleted the CD8 T cells, and they saw the virus bounce back transiently. So they argued that this was CD8 depletion in the setting of very early antibodies. And again, the concept is if you give these antibodies very early during viremia, there's a ton of virus and a ton of antibody. You get these immune complexes. They're able to engage with the immune system and hopefully, theoretically, boost these T cell responses that without that stimulation wouldn't have been able to control virus. So this is sort of the first inclination that early antiretrovirals plus BNAbs might have an immune modulatory effect that could lead to virus suppression. Okay, the next study was just published last month, and this is by Dan Baruch's group, and this is looking at, again, very early treatment, but looking at broadly neutralizing antibodies plus a latency reversal agent, in this case a TLR7 agonist, plus antiretrovirals in very acute infection. So what you can see here is they had 4 groups of animals, the antiretrovirals alone, antiretrovirals plus the TLR7 agonist, antiretrovirals plus antibody, or antibody and the TLR7. And I don't know if you can see here very well, but this is weeks of infection, so they actually started both antiretrovirals and these therapies at just 7 days after infection, so this is very, very early. You can see the viral loads come up and they come right down. And then they maintained this antiretroviral suppression for more than a year, and then they went ahead and they performed treatment interruptions, so they stopped the antiretrovirals. And in the sort of sham animals, you see virus rebound that looks similar to what we see in HIV-infected individuals who have their treatment interrupted. But down here with the antibody plus the TLR7 agonist, you can see there's a significant reduction in the number of animals who rebound and the magnitude of the rebound, and then many of them go on to control. And it's interesting because in a subset of these controlling animals, not only was virus suppressed, but when they performed a very rigorous test to see if there's any virus, any replication competent virus left, they basically did adoptive transfer. So they took a lot of tissues and blood and they transplanted that to another animal, and they were unable to find new infection. So this suggests that not only was virus suppressed, but they were actually able to clear much of the virus-infected cells from this animal. So in this very idealized system, I think that there's early evidence that very early BNAbs could lead to reservoir suppression as well as clearance. But there's very important caveats to these studies. The first, I think, and there's a lot of nuance when we talk about all these animal models, but these particular models of SHIV infection do not truly represent the sort of rigorous virus challenge that HIV-infected humans encounter with their virus. I also think that ART in hyperacute infection is really important to consider. It's, first of all, something that we cannot do in HIV-infected individuals because no one is diagnosed before they even have sort of symptoms from early infection. And this leads to a much smaller reservoir or a much smaller challenge that these antibodies and other immune factors have to overcome. And I also think the mechanisms involved are not completely borne out, and so we need to do a lot of mechanistic studies to understand what happened in these animals as well. But this leaves us to human clinical trials, and I'm not quite as cool as Dr. Blankson, so I don't have Miles Davis, but this is Bjork talking about human behavior from a while back, so we talk about human clinical trials. So the first human clinical trials were published in 2015, and basically looking at infusion of a single antibody in viremic individuals. This is a study by Rebecca Lynch and her colleagues at the VRC. And basically, they gave to 8 viremic individuals an infusion of this CD4 binding site antibody. Again, they saw an antiviral effect with transient reduction in 6 of 8 individuals. But importantly, in the 2 individuals who did not see much viral decline, there was found to be high levels of baseline resistance to this virus. So this is one of the first things that's identified when you move from a macaque model where you have a very well-characterized virus challenge that you know is sensitive to your antibodies to human beings where we don't know what lies underneath. Okay, so resistance was identified in these 2 individuals, and a resistance arose in the other individuals who encountered this antibody in the face of ongoing virus replication. All right, the nuisance-wide group also did a very similar study looking at 3BNC117, which is a modestly more potent CD4 binding site antibody. Again, they saw transient suppression, and they saw resistant virus emerge. So the next set of experiments were looking at whether a single BNAb could maintain virus suppression after treatment interruption. So theoretically, this bar was a little bit lower than suppressing active virus replication, and we thought maybe maintaining after treatment interruption would be a little easier. So the first set of trials, there were 3 trials looking at 2 different antibodies, BRCA1 and 3BNC. They all followed a very similar format to chronically infected individuals, gave them 3 infusions of broadly neutralizing antibodies, interrupted therapy shortly thereafter, and followed them very closely to monitor for viral rebound. And this is the Kaplan-Meier curves looking at time to rebound for VRCO1, the 2 studies, and 3BNC. The historical controls are represented by this blue line and this sort of jagged black line. This is actually exactly the same data, just represented in slightly different ways. But what you can see is each of these antibodies alone was able to slightly push this curve to the right, delay the time of rebound a little bit, but it was pretty modest. So in VRCO1, we saw either 4 or 6 weeks of as a median time to rebound, whereas with 3BNC, we saw either 6 or 10 weeks, depending on the route of administration. So the VRCO1 trials did not do any pre-screening for resistance. The 3BNC trial did, but in all studies, they found high levels of pre-existing resistance, and also certainly in the VRCO1 trial that I was involved with, when we characterized the virus from these early rebounders, what we found was looking at virus that was from the plasma before antiretrovirals. I'm showing here IC50s to VRCO1, and you may not be able to see this very well, but actually these numbers are pretty high, basically meaning that in our early rebounders, we had very high levels of baseline resistance to VRCO1 before they'd ever been exposed to the virus. So this is sort of, again, resistance becoming a major issue. So the most recent iteration of these trials was just published last month by the Nussensweig group, and this was looking at 2 bNAbs, the CD4 binding site antibody 3BNC and 101074, a V3 antibody, given again in treatment interruption. And this is sort of one of our first sort of positive iterations of this study. So the same sort of situation, except people in this study were pre-screened using a bulk outgrowth assay to look for resistance to either of these antibodies. So they used a cutoff as an IC50 greater than or equal to 2 micrograms per mil, and they actually excluded more than half of potential participants because of this threshold. They ended up enrolling 15 individuals who got 3 infusions of these 2 antibodies and were followed closely for viral rebound. In the meantime, they also did a more sensitive assay, doing basically a limiting dilution virus outgrowth assay to say, if we look at a bunch more reservoir viruses, can we identify any resistance virus? And unfortunately, 6 of the 15 enrolled participants using that more sensitive method were identified to have resistance. So treatment interruption was started at day 2 after infusions. And what you can see in these Kaplan-Meier curves is if we look in the middle at those 9 individuals who had sensitive reservoir, the virus curve, or the time to rebound, is actually pushed substantially to the right. So there's actually a median of 23 weeks of virus suppression in individuals with sensitive virus. So this is a pretty positive outcome. If you have a sensitive reservoir, we're able to maintain a reasonable duration of suppression with these 2 antibodies. However, if you look at the 6 individuals, this is just looking at 2, but if you look at individuals with resistant virus, the ability to suppress virus with these 2 antibodies is very limited, and reflecting the fact that the resistance in some participants is really going to be a limiting issue. So I will say this is looking at the individual virus curves, and if we look at 2 of the individuals who had sensitive reservoirs, it's very interesting because 2 of these individuals, in addition to having suppression while the antibodies were in circulation, actually had maintained suppression for long after when those antibodies waned. So it's been about a year now since this study started, and Marina Kaski last week at the NIH meeting presented the fact that at this point in time, one of these 2 participants has just rebounded, so it's been about a year, and the other individual, the one here who has 0 little blips on their virus viral load assay during this trial, and has had really no detectable virus for almost a year, maintained suppression. This individual who had some blipping had the year-long suppression. So this is a small study without a placebo arm, and it needs to be taken in with all of those caveats, but it is exciting to see that there is some long-term post-treatment control in some individuals, and something that we're going to have to dig in to see if this is just, statistics, or if this is a unique effect from immune modulation with these antibodies. So I think in life it's very important to join the resistance and think about that as in our trying times. However, for this purpose, we're going to try to fight against the resistance of antibodies, and I just want to talk extremely briefly about some of the work we've done to try to think about how can we do a fast and accurate and sensitive screen for pre-existing resistance before we enroll people in these trials. So we partnered, we meaning many members of the ACTG, partnered with Monogram Biosciences, which is a company many of you have maybe worked with if you've done any clinical medicine, because they do a ton of screening assays, the same basic assay platform to look for antiretroviral resistance. So they have a platform called PhenoSense NAB, which basically attempts to reproduce the virus diversity in any sample within this assay. So they basically take either plasma or PBMCs, and they make a swarm of the envelopes that are represented within that sample, and they co-transfect that with a luciferase-expressing backbone, and then through this high-output sort of NEWT assay can test sensitivity to that virus population against a range of antibodies. So what we wanted to do was to look at this assay and see how it performed against a relevant group of patient samples against antibodies that are currently in clinical development. So through our generous partners at the VRC, including Lucio, and at the Rockefeller, we got a bunch of antibodies characterizing these 4 classes, and we tested them against samples from 65 participants of a recent ACTG trial. From each of these participants, we had a pre-ART plasma sample, and then PBMCs from 1 and 3 years of ART suppression, and our goal was to sort of assess how did this assay perform in those 2 different sample types, and how did it perform in this recent and relevant clinical group. So I'm showing you just VRC01 data, but each one of these little dots is sort of the mean sensitivity, it's not sort of, it is the mean sensitivity of one patient's virus population against each of these antibodies. So the red is looking at pre-ART plasma samples, green is year 1 PBMC samples, and blue is year 3 PBMC samples. And to me, the salient findings are, first of all, for all of these antibodies, there's a huge range in the baseline susceptibility from patient to patient. And this is a really important thing to think about when we are giving one antibody to a patient. We could have a whole different story depending on that baseline resistance. The other thing I think is interesting is if you look at the pattern, the distribution seems to be very similar, going from one sample type and spreading over 3 years of infection. So if we do correlations between, here's looking at year 1 PBMCs and pre-ART plasma, we can see that it's actually a very nice tight correlation. And so we think that we can generate very similar data from a sample collected from a participant who's been on ART for a long, long time, and we don't need that pre-ART plasma value in order to be able to get some of this information. So this is looking at the pre-ART plasma across all 7 antibodies. And again, I just want to emphasize the huge range in each of these antibodies' susceptibility from one patient to the next. We can see sort of the relative potency of these different antibodies, which is fairly representative of what has been published by other means. So that's reassuring to a certain extent from this, this study. And finally, what we wanted to do, and this is just the very first step in validating this assay, because we think the assay's characteristics in and of themselves are good, but we need to make sure that they work within clinical trials. So we took this assay and we performed this sensitivity measure on PBMCs from entry to A5340, which was this VRC01 and treatment interruption study. And what I'm showing here in these green dots is comparing the Monogram PhenoSense assay against our in vitro assay, where we basically cloned single genome sequencing derived OMS. And what you can see is there's a very tight correlation between resistance that we detected and resistance that the assay detected. So that is reassuring and was statistically significant, despite the fact that we just had 5 participants with available samples. Finally, we wanted to look at the time to rebound in this trial and say, did this pre-screening method predict whether this antibody was going to be effective? And if we do a Spearman correlation, we find that it is not, there is not a good sort of indirect correlation here. But, and this is me and the statisticians in the room, please cover your ears. But if you look at these numbers and you sort of squint, you can sort of see, if we take a cutoff here, if you're more resistant, you're more likely to rebound very early. If you're more sensitive, you're more likely to rebound very late. So we don't have enough data to say anything, but I do think that these are encouraging data allowing us to sort of validate this in future clinical trials, and that is our plan moving forward. So this assay is going to be used in several ACTG and several of the Delaney Care Collaboratory antibody-based trials. And I'm hopeful that it will be helpful for the trial, but also allow us to validate it in a real setting. Okay, statisticians, you can take your hands off your ears now. So, in summary, I think that these novel BNAbs with enhanced breadth and potency, they've been identified, and they're currently, knee-deep in studies for prevention, treatment, and cure. And it's going to take these empiric studies in humans to really figure out what their worth is. There's a lot of theoretical promise, I think, for this sort of being the kill portion of a kick and kill strategy, or being able to maintain suppression for a long period of time because this is a safe and long-acting modality. I think there's really interesting hints of utility in non-human primate studies, but we're going to need more robust animal studies, mechanistic studies, and human clinical trials to really bear those answers out. Our human trials are still in early phases, but the promising results in those individuals with sensitive virus, I think, are exciting. And you can ask Lucio how much energy there is to try to do these, as his phone probably rings off the hook every day with people wanting antibodies to test their antibody plus X intervention. Anyway, there are clearly challenges that still remain. I think resistance is going to be very challenging. The cost and pace, et cetera, the need for effective LRAs, these are all important. So in conclusion, I think in my mind, you can either say that the glass is a quarter full or 3/4 empty, but at least there's water in there and we're moving in the right direction. So that's my sort of like hope, hype, etc. And I really think the next few years are going to be exciting as we figure some of these things out, and it's going to take a lot more time for us to figure it out. Probably way over time, so thank you very much for the opportunity to talk.
Thank you, that was outstanding. In the interest of time, I might ask if all of our wonderful speakers could join us at the desk here for the panel discussion. I'd also like to introduce Dr. Tom Quinn, who agreed to moderate our discussion. If we could get a separate microphone for him. Anywhere. I think anywhere is fine. Yeah, Dr. Quinn is the...
Go for it.
Like the middle.
Do you want to moderate from here? Or she's gonna... Yeah. Dr. Quinn is the head of global health at Johns Hopkins and also an intramural investigator at the NIH. He was instrumental in characterizing the global HIV epidemic. I'd also like to invite Drs. Silicano and Ambinder to the panel. Dr. Silicano really doesn't need much introduction since he's been mentioned multiple times during this session. Dr. Ambinder is a professor of oncology here who has also significant interest in transplant-related approaches to curing HIV. And with that, I'd like to...
Thanks, Ashwin, and welcome everyone. What a great day this has been. All the speakers deserve a round of applause for just great presentations. As Ashwin pointed out, and it's been mentioned a couple times today, there was a meeting just last week for 3 full days at the NIH. This is all online. If you want to hear Tony Fauci's talk or you want to hear Bob Silicano's talk, you can access all of them. So they've historically have been saved. And after 3 days there, I questioned myself, why did I even bother going when I could have just done today and learned just about everything that I learned for the 3 days down there. It is absolutely amazing how much progress has been made over the last probably 3 to 5 years. We really, I'm going to pick up on terms. Deborah Persaud first said it, hype and hope. And I do think it was a lot of hype, but we've moved into the hope arena that someday we may be able to at least prolong a durable suppression more than 1 year. This durable suppression of a month or 2 months is really insignificant in the bigger scheme of being treated for 45 years. What's 1 or 2 months going to make here and there? So we really want durable suppression over a year, and we're not there yet. We heard a lot of exciting data in the primate models, even in today as well. But I think the title of this moderation, and I'm going to open it up to lots of discussion from all of you to ask the panelists questions or the panelists to argue with one another. I always like debate. But the title of this, this discussion is really a reality check. Where are we? And I'll start out with a couple things. Lynda Dee mentioned that we're spending $157 million a year now on this. And that's probably 1/5 of what we're spending on prevention. Is that the right balance? When we have 1 million people becoming infected each year globally, and we have 22 million people actually on treatment, what is the reality check? In the sense that when we use that term, start thinking about cost-benefit ratios. Are we going to be able to afford infusion of BNABs? Because you ended at the end, Katie, with how expensive these are and production of them compared to that 1 pill a day. Costs are different. Where I work in Uganda, it's $350 a year. In South Africa, it's about $400 a year. Here in the U.S., it's about $10,000 a year for that 1 pill a day. There are cost differentials that we have to take into consideration. So keep that in mind as we go down the panel and ask questions. And I'm going to start, I'll get it going. I like debate. And I like trying to be controversial. I will say, Katie, I'm always a glass half full. And so I do, although it won't sound it as I ask these questions, I do like to be on the hope side and that we're making the progress. So please don't take some of my comments as the pessimist of the group. I'm really much more the optimist. And as Bob Silicano knows, we've already launched some cure initiatives in Uganda and then South Africa. And I'd like to see more international development in adults as well as there are in the children, which Debbie pointed out. But let me come back to Jeremy and Lynda. Where's Jeremy? I can't see. There he is. Short. We'll start out from the community level, the community of HIV-infected people that would like to be cured. And during lunch, we started talking about one of the underlying issues is really stigma, that people who have HIV and take this pill a day to be virally suppressed still live in a life of stigmatization. And a cure would help deal with that. It would help with decreasing transmission, although if they're virally suppressed, U equals U, that in the background is when you have to think people become non-adherent, as we heard in some of the examples. So again, Lynda and Jeremy, just thinking about the cost of all this, the issues of the population at risk, how difficult, what are the struggles we need to deal with now to make sure these trials move forward, but they keep in mind the benefit for the individuals and the cost to society overall? So either one of you can jump into that.
So Jeremy, should I go first?
Yeah, go ahead.
Okay. So I think I talked about this morning the impediments and the different sort of points that you made. I talked last week at the meeting about the cost of the BNABs. And also, what I didn't say was, it's how realistic is it to, and I'm sorry to pick on you, Katie, to think we're going to find people 7 days after they're infected. That's when these drugs are the most effective. But for me, I tend to be really practical. You're going to have to spend money. I mean, when we started AIDS Action Baltimore, it wasn't 6 months that before we said, you know what, we're never going to get away from this unless we do research. So we're going to have to pay for things, and we're going to have to find the money for it, and too bad. That's where and that's what people like me do at the NIH and, with Congress. It's harder now than it was. I mean, everything's going to Alzheimer's. They've got our money. But anyway, so I'm going to try and get some of it back. But anyway, and not just me. But, I don't look at it like that. For me, the most important issue in the beginning is to measure the virus, if we can't do that, we're going to have to continue with the ATIs. We're going to have all these ethical issues and all the risk that comes with that. And for the initial sort of early stuff, that's the most important thing for me, And maybe I know too much about science. Maybe I'm not, You know, I mean, community people oftentimes are into social justice sort of things, but to me, social justice means getting this done, getting this work done. And the best you know, path that I see to getting started to this and getting a real handle on this sort of cure issue period is to be able to measure it. You know, just like the viral load from years ago. Until we have that, we're like shooting in the dark. You know.
Jeremy?
So thanks for the question, and your intuitions are correct. So the early social science work that's been done among participants in various HIV cure-related research studies indicate that in Southeast Asia and the Thailand studies that are doing a lot of early treatment work, that the number one reason people participate is to hope to decrease transmission to others, actually, and the second one is stigma. And a lot of that work hasn't been reproduced in the North and the West, but you're hearing some very strong stories about the desire for a cure is not solely personal. It's deeply social, and it's a social phenomenon which is hard for us to imagine. It's not part of the science toolkit. Now, the other questions you raised, though, Tom, are justice kind of questions about the appropriate allocation of resources, which we've been discussing since early on in the epidemic. Now, if you remember early on, the big discussions were how much should we put towards prevention and how much should we put for treatment.
Right.
And those discussions have not been settled, and justice is the hardest set of problems to crack within the allocation of science funding, and who gets to decide raises huge and inexorable challenges. Aristotle messed with it, and he didn't figure it out. He was a pretty smart guy. And we've still been struggling with justice questions since then. But I think one important thing that we've done to advance social justice is to try to say what's the extent to which community input is important, scientific input is important, and policy expertise is important. And without considering that from multiple spheres of knowledge, you're not going to get closer to a right answer of what constitutes fairness. I'm sorry for the ethics lecture late in the day. Look, no slides. And but the piece here about the justice consideration is, yes, eventually we'd like something that was scalable, and none of the things we're doing right now are scalable, right? But that's not the state, that's not why we're doing these science. So any of these things, I mean, the infusions that are being done in parts of Africa related to HIV prevention with BNABs, huge, gallons of stuff being infused, and people are lining up because they want to contribute. But that's never going to be a scalable solution. A lot of these things are not scalable, but that's okay, because it's what we're learning in the process of this that'll inform something. We can worry about scalability later. So I think we have to let the science drive some of those decisions and not get in the way of the fact that we can't pay for whatever it is now. We can bring prices down. If you had told people prior to George Bush's presidency that there would be buckets of antiretrovirals delivered to Africa, no one would have believed you. And in fact, we have this thing called PEPFAR where lots of people in the world are being treated. Not everyone, not enough, but it was a big head start.
Oh, huge response with PEPFAR. Debbie, jump in. You want to add to this?
Yeah, I wanted to add because as an advocate for pediatrics, I think Lucio could tell me how many pediatric patients we could treat with one infusion being used for prevention in an adult. I think it's a 50 or 100 with one infusion because they're only 2.5 kilos. It's 40 milligram per kilogram. It can be given subcutaneously. It can be given in sub-Saharan Africa. We're showing that it can be given in sub-Saharan Africa. And also with perinatal HIV infection, we have that window of opportunity. We know when kids are exposed. So we can give within 2 days, within 7 days. I mean, I think the situation we face, which is what Jeremy raised, is really the risk-benefit and a favorable profile. So, for example, in perinatal infection with prevention strategies, the transmission rate's about 2%. In untreated women, for neuroinfection, about 9%. So that translates into exposing 90% of potentially uninfected infants to neuroagents. And that's the sort of decision-making we have to think about. But these infants that we're enrolling in these first proof of concept studies are high-risk infants. They're high risk for peripartum transmission. They're high risk for breastfeeding transmission. Mary Glenn could comment on this. Accounts for like 50% of our transmissions now that we know how to prevent infection. Breastfeeding is life-saving. So there is, in my mind, a very clear pathway and clear rationale for doing these studies in pediatric populations. It sort of mimics the PMTCT. Women and infants were the first set of individuals to show us that ARC can prevent transmission and post-exposure prophylaxis can prevent transmission. So we are hopeful, Tom, with all of the emerging data with early treatment, BNABs, and the data that Katie showed with combination, that we can affect remission in this population.
Well, and I think your comment in the data, which Bob also picked up on, looking at the lifetime survival of children who are perinatally infected or infected in early adolescence, is much shorter than their counterparts as compared to adults, where it's pretty close right now. I wanted to move the discussion along. Oh, Katie.
I'm sorry, I just want to make two small comments. So
Yeah.
I do think this cost benefit is a very important issue. We have limited resources. There's so many important things just within HIV that we need to deal with. But when speaking about cure, first of all, if we could at some point, and it may seem like science fiction now, but if we could at some point have a one-time cure event, you have to balance that against 40 years of antiretrovirals or 40 years of therapy. So the scale, it's not it costs this much a year versus this much a year. It's you have a one-time cure versus a lifetime of therapy. And I just want to say in defense of, I'm a homer for antibodies, but the goal is not to give boatloads of infusions to everybody. The goal is to figure out what works and then figure out how to deliver it in the most cheap and long-term sustainable way. So in theory, in the future, we decide this is the concentration of antibodies that we need in this setting. We have a vector delivery. You get a one injection, you've got 10 years of those antibodies. That is a much more cost-effective ultimate solution. There's a lot of hurdles to get there, but we're not thinking about trying to deliver, extremely expensive things every 2 months, ad infinitum.
So, Katie, I'll just give you sort of my example of your argument, which I used to use all the time with male circumcision in Africa. For $15, it's a one-time 15-minute surgical intervention, and that person has a 60 to 70% protective effect, which is comparable if you look at all the PrEP trials and the cost of PrEP. And thanks to PEPFAR endorsing that for Africa, we have circumcised 15 million men. So it does take off. And so I agree with you on if we can get to that point. So far, we're at durable suppressions of just a few months. And we need to get beyond that. And I think the science that everyone's working on will get us there. So I'm on that road of hope that we will make it to that.
Just remind people of one thing real quickly.
Yeah.
Cost benefit, Gilead, HCV, okay? That's their argument. Look what happened when they charged a little bit, just that was like $10,000, all right? As opposed to, that's just part of the regimen, the BNAB. So if we don't have something that's a lot cheaper than that, people don't look at, we don't do things like the rest of the people in the world as far as our health service. We don't have a health service. The government doesn't have to pay. So people look at this as one quarter at a time. They don't look at the long-term effects of this. So Gilead killed the goose that laid the golden egg with their HCV cure for what they charged for it. So, if ours is like 3 times more than that, we're not going to have anybody paying for it.
So I'm going to take this discussion up another notch and start getting to some of the science parts. Lynda mentioned if we don't have a way of measuring the latent reservoir, we're really up creek. We're not going to really make much advances. And so since Bob has joined us, and Bob's lab has been instrumental in the development of the gold standard as well as some new assays that are going to be much more practical for measuring changes in the latent reservoir, I want to give Bob a few minutes to explain his perspective on measuring the latent reservoir. And then next to you is Rich Ambinder. When you're done, hand it to Rich to tell us what he's been doing in terms of the cure effort, because we didn't get to hear about that.
Well, thanks, Tom. I guess I'd like to take issue with something that Lynda said, although I hate to do that after that really absolutely brilliant talk that Lynda gave. I mean, I think we do have ways to measure the reservoir, and we've actually had it really since the beginning. We can measure replication-competent virus persisting in patients. But as people who've done this assay, Janet and Joel and Christine and Janice now in SIV, it's a very difficult assay. What most people do is simple PCR, and that's what's being used in most of the 90 or 100 clinical trials that are going on. That assay is absolutely the wrong thing to do because it's mainly detecting defective viruses which are unlikely to be affected by the intervention. So one of the things that we've done, largely through work that Greg Laird has done and others in our group, is to develop a simple, scalable assay for the non-defective viruses that have a potential to cause rebound. And we hope that in the future that this will be a sort of a better way to see whether we're making any progress. Rich?
So in terms of hype or hope, I think the devil's in the details. There's been huge progress in bone marrow transplantation in general in the last 15 years, and our ability to transplant people and get them through transplant has profoundly changed. In addition, it's now straightforward to find CCR5 Delta 32 donors for most Caucasians and for a substantial fraction of African American patients. I'm quite sure that within the next 2 years, the number of HIV patients who have been cured through transplant is going to go from one or, as Debbie said, maybe two to date to several hundred. But transplant is still a nasty thing, and with all the dramatic improvements, it's not something that's appropriate to think about applying to the general population. But I think in a very special subset of people with hematologic malignancies or other indications that they need an allogeneic transplant, I suspect we'll be curing 2/3 of people in the very immediate future. I think the major barriers have disappeared. I'm not sure how we make the next step and extrapolate that to other settings.
Yeah. What do you think about, there's the group at the Collaboratory out in Seattle working on CAR T cells, and which are great in cancer as well, but they're now trying to apply that for HIV. The results that they showed weren't real exciting, but they're trying to make some progress. Do you want to make a comment? Because we didn't have a presentation on that, and that is the genetic immunomodulatory effort to cure HIV.
So CAR T cells are fascinating when Lynda Dee talks about the price of therapy. So CAR T cells are now approved for lymphoma. We are participating in trials in lymphoma and in standard of care CAR T cell treatments for lymphoma. It's $450,000 a shot. And so transplant is a bargain in comparison.
Think of that.
As it is in comparison to a great many other very expensive but very effective new cancer therapies. And so the whole economics of how we charge for drugs is, I could only discuss over a drink, and I'm not the expert. I only see the price on the menu, which is truly out of sight for many treatments. I think the limitation of the CAR T cell approach is going to be what the antigen is that you're going to target with the CAR T cells. And so I don't think they're the be-all, end-all. I actually expect them to be much less effective for HIV than for cancer. For HIV with allogeneic transplant, using the Silicano viral outgrowth assay, Christine and her colleagues have shown that, and other people at other places, everybody who gets complete donor engraftment, where the donor cells replace the host cells, you cease to be able to measure HIV by the assay. Now we know from the Boston patients and from one of our patients who was non-compliant that those patients aren't necessarily cured and that they will rebound, but there is no intervention that is more effective in 2018 than allogeneic transplant in terms of being able to get rid of the reservoirs that we know how to measure. But CAR T cells, like antibodies, are going to be very interesting, and I think we're going to see a lot of incremental advances, an explosion of different technical approaches that together are going to start curing people. We're going to nibble at the edges for a while, but we're going to be curing people.
So using the latent reservoir measurement assays, IPDA, which Bob's lab's getting into, and it has developed QVOA, which has been the standard. Moving it to get the other panelists involved with both the primate models. Has there been any, in the mouse model, Liang, have there been any successes in reducing the size of the reservoir? We did hear a lot on durable suppression in the primate models. What about reduction in the size of the reservoir with either SIV or SHIV or what have you? Guido, that's aimed at you a little bit. And then Janice.
It's a good question. Thanks, Tom. I think one of the complexities here, and I try to embrace complexity rather than being, afraid of complexity, is that the non-human primate model in reality is many different models, and there are different viruses, there are different monkey species, there are different type of inoculation, there are different approaches to antiretroviral therapy. I guess the one thing that, from the point of view of the applicability to human studies, that I think the field should think about seriously, is that there's been a lot of high impact studies that have been influential in the field using systems in which the animals were infected and then treated with antiretrovirals very, very early on. Yeah. Day 3, there was an influential study by Nishimura and colleague in Nature by the Mal Martin and Michel Nussenzweig teams. Day 3 post-infection that were put on ART. There's another highly influential study by Dan Baruch that was published with James Whitney a few years ago, also in Nature, with the animals treated at day 3, 7, 10, 14. Nature Medicine paper by the group of Louis Speaker that was initially designed to be a study of the CMV therapeutic vaccine that turned out to be a study of early infection. And that study, also the animals were treated, between day 3 and 7 post-infection. So I think it's that's what Liang and I were sort of joking about, on the shuttle this morning. If you remember, there's this idea that validating a model equal publishing in some high-impact journal. It's not always the case. And as a matter of fact, I am not too excited about those models where the animals are treated, on day 3, 4, 5, 6, 7 after infection, because A, it's not very relevant to what you can do in humans, number one. Number two, you perturb the system in a very, violent way. You have immune responses that are completely different than what you see normally. There is no maturation or aborted maturation of the CD4 responses or the CD8 responses or the B cell responses. There's this weird sort of artificially low and possibly skewed reservoir in terms of both size and location and cell subsets that are involved. And then things might work, and then, you know, the challenge is, okay, what if you now treat animals at 2 months after infection or 6 months after infection? Is that, you know, going to be there too? So I'm actually afraid that there's going to be a lot of excitement for studies in which the animals are, I'm not saying cured, but, you know, put into some kind of long-term remission in those artificially low bar, you know, type of experimental settings. And that's why actually our team, you know, likes to at least let them go for a couple of months. I think it's important for a point of discussion. I don't know what Bob and other leaders here, and Katie and others think, but I think it's something that we as a field should discuss a little bit more, you know, more awareness of the potential issues.
Janice, you do a lot with primate studies. I know the focus has been on macrophage infection, which I have 100 questions about. But in terms of what Guido was stating and perspectives on using the models as an initial understanding of the science and before going into the humans,
Right.
besides safety.
So I think when you get back to the issues of money versus science, that's always an issue. I mean, when we put in a grant, we have to consider how long we're going to take to do the studies, and every day costs you $10 or more for a primate. Yeah. And so I think that is the balance. So you try to get the model as close to what you think it might be. So in the sense, we've used two models now. The dual inoculated model actually was meant to circumvent that a little because it put a very large swarm. We put in 2 swarms of virus. And so the animal was infected, and then we did treat after 14 days, but we could actually see very different viruses, and we could track back to whether that was inoculum virus or whether it had evolved from the spleen or the brain or other things. So, so there's ways of, now people don't get infected that way, but there's ways to use the model to then make up for that. So we have to remember they're models, and we actually don't know how each person gets infected. And each person's probably very different. And so the model, you have, as a homologous a background as you can, right? We have these monkeys, which we eliminate certain genes so that, and so it is a model. And so I think what you do when you infect, when you look, depends on the question you're going to ask. Yeah. And so for us, when we're looking at whether macrophages are infected and where the reservoirs are, then I think, we have to be careful and infect long enough or with a diverse enough swarm. And even the SIV 251 swarm is a swarm. But we found that very early, we did experiments very early in the dual infected model, and by 4 days, we actually sacrificed animals at 4 days. The brain was seeded and all the tissues were seeded. Everything was seeded. It was, IV blood, but we did check that. So it really depends on the question you're asking and what you're trying to find out. And so we've focused on the disease process and now latency and the location of different cells. And as I said, we actually had been, for a long time, nobody had done any QVOAs in the SIV model. They're very expensive, so you add that on. I mean, recently someone did report it, but a very limited QVOA. I think if we want animal models to be models for HIV, we have to investigate at the level we do in people, what's the size of the reservoir, what kind of reservoirs, and in the animal model, we have the opportunity to look at the tissues.
Yeah.
And that's something you can't do in people. You can't look at the different tissues and figure out what's going on there.
The other thing that took place at the NIH last week was a lot of sequencing of the virus evolution, changes in the reservoir, changes in the cell types, and so forth. And I didn't know if you had done sequencing of the virus from the macrophages versus what you recover from T cells.
So we did a lot of sequencing back in the early 2000s and showed that virus evolution does contribute to the virus that causes CNS disease. So the early virus that gets into the brain is not the virus that we would find in neurological disease, and that it evolved from the spleen and other tissues where the macrophage tropic virus got expanded, and the tropism for the brain then came from. So we've done that. We continue to do it. We've looked at, we did an experiment in which we used an LRA, and one of the animals that was suppressed for a year and a half did rebound on the LRA. Now, we don't know it was the LRA because it was only 1 out of 3 animals, but then we sequenced those viruses, and what we showed from the sequence is that the CSF and the plasma actually reactivated on the LRA, and they were 2 distinct viruses. So there was compartmentalization of the virus that reactivated in the brain, and there was compartmentalization of the virus that reactivated in the. So we have done those studies too. So I think with a non-human primate, you've got to decide what your question is, and then make sure the model and the timing and everything else is answering your question.
Let me open it up. I've got other questions if no one else wants to ask any, but just raise your hand. We've got microphones. So, Greg.
Yeah, so I had a question for Katie about susceptibility testing to antibodies. So there's sort of 2 questions. The first was, knowing what you do about what Monogram is doing and what people are working on, how deep do we have to look in patients that are going into these trials? And then the second sort of related question is, where is the resistance coming from? Is this acquired resistance that people are getting from the person they're infected by, or is this resistance that's developing because of the antibodies that they develop? And the corollary of all this is, how big of a problem is this going to be for implementing these studies and finding people where we can actually treat with combinations that will be effective?
Okay, thanks. Great questions. I'll try to be fast. But I probably won't be. So the second question first, where does this resistance come from? Well, all of these broadly neutralizing antibodies, basically I think every individual in the course of their infection develops some degree of resistance, and that virus then gets transmitted to the next person, et cetera. So, if we think about broadly neutralizing antibodies, they are targeting epitopes that many strain-specific antibodies from any one individual are also targeting, maybe not in a broad manner. But anyway, so as any individual virus population responds to the autologous antibody response that they happen to encounter, they're going to develop, resistance to this, that, or the other. So if we look at transmitted founder viruses, if we look at viruses in chronic infection, there's just whatever that virus has been responding to over the past however many generations, and that person and the host before, they're going to accrue different patterns of resistance, including resistance to broadly neutralizing antibodies, even if that host didn't make a broadly neutralizing antibody. That was a convoluted way of saying I think that these things accrue over a long period of time, and there isn't necessarily such a high fitness cost to these sort of resistance that we don't think that these sort of things are possible. And can persist for a long period of time. So I think that in terms of your first question, how deeply do we need to sequence? I think that's a really challenging question. I don't actually think we're going to be able to develop a foolproof assay that's going to be able to plumb the depths of the reservoir in any particular individual. But if we're trying to make we have to work on both fronts. We have to try to make antibodies that are as broad and potent as possible so that we think, just in general, they are the best versions of targeting whatever epitope we can, so that there is a lower prevalence resistance just everywhere. And then we have to look with whatever, the best methods we can within whatever sample is available. So, logistically, if we're doing a trial and we have a bunch of chronically suppressed individuals who are willing to participate in that trial, what can we ask of them? We can ask of them to maybe give us a sample of PBMCs. So that's a large blood draw so that we can take those cells and we can look at what the peripheral cells have as integrated virus in whatever version we want. But that's kind of the most I can think we can ask of a trial participant at this point in time. And if we can plumb the depths of that peripheral PBMC integrated virus sample, whether it's replication competent or not, we can do as much as we can with that sample, but that's kind of the extent that we have. Is that going to be able to detect all the resistance in that individual? I think absolutely not. But I think that we're going to have to be able to exclude those participants who have superficial resistance, and then we can make sure that if there's no benefit for that antibody for them, that they will not be in the trial. But we're going to have some individuals that still have resistance who come into these trials and don't benefit from these antibodies. And then we just need to keep coming up with better antibodies, better combinations of antibodies, so that hopefully the two things together, screening for antibodies that don't work and then having as broad a coverage with potent antibodies on the other side.
Ashwin.
I had a question for Joel and for Guido. It was really very interesting to see the CD8 work. Is there a way that you can envision training or educating CD8s? On the one hand, we see the BNAB therapeutic approach really progressing quite far. Is the only hope to get commensurate success with CD8s a vaccine strategy or CAR T cells, or do you see an intermediary step where you can somehow train CD8s? For instance, I think the post-treatment controller story is really almost emblematic of the hype versus hope dichotomy, in that you see a large number of people who are post-treatment controllers after the first year of interruption, but then that diminishes pretty considerably. How would you consider exploiting the CD8 story a little bit more for therapeutic benefit?
Well, you think of the answer there. I also had almost the identical question, but I picked on one of the words that Guido used, but Joel, you also implied. That's transcriptional silencing versus CTL, and that CD8s are playing a role in both. How can they do both? How do you really get transcriptional silencing? That was your term, Guido.
Yeah, no, I think we have to be, it's like if somebody asks a CD4-ologist, how can CD4 cells do Th1 and also do TFH and also do Th17 and also do Tregs? How is that possible? Well, they do it, right? We like facts in science, and we know that they do it. So I think we shouldn't, cut the CD8 short and tell them that they can only do one thing because thou shalt only do CTL activity and nothing else because so God decided. So I think the important thing, Tom, is to really understand, those subsets of CD8 cells, what are they doing, how can we this thing. I think we're kind of getting there. I didn't want to say too much because it's preliminary work, but it looks like the transcriptional silencing is mediated by a subset of Treg-like CD8 that are producing IL-10 and TGF-beta. The CTL we know is mostly related to a subset of CD8 cells that are polarized in the production of perforin and granzyme B through TBAT activation, and also make TNF and interferon gamma as predominant cytokines, some of IL-2 as well. So I think we know that there are different subsets of CD8 with the different phases. So if we can, for instance, magically figure out a way to make the CTLs much stronger and maybe those pro-latency CD8 much weaker, we could have a major impact on the reservoir size. And I actually think that that's one of the biggest hopes that we have in the field at this point. We just need to, face this problem in a sort of open-minded way, fact-driven, data-driven way, not look at pre-existing paradigms as the only thing that we can deal with.
Joel?
I think CD8s are tough because of HLA restriction. So we know certain epitopes are conserved and are targeted by CD8s from elite controllers. But if you don't have the right HLA, your CD8s are not going to target that epitope, and it's hard to get around that. Now, CAR T cells are nice because you can move around MHC restriction because you're not looking at a peptide in MHC. The other thing I like are the bispecific antibodies, where one arm targets an epitope which is not MHC restricted, so like an antibody to envelope, and the other triggers CD3. So you can activate T cells and will kill infected cells. And I think that has a lot of promise. It's a lot cheaper than CAR T cells, and it can get into lymph nodes and B cell follicles where CD8s can't. So I think there's promise for that approach.
I want to add it
Can I make I'm sorry, just a very quick comment. When we talk about class 1 restriction, I think it's important to remember that there are two levels at which CD8 cells are class 1 restricted. One is the level of recognition, initial recognition, which means that they get activated, and then there is the level of effector mechanism, which applies particularly to the killing of an infected cell expressing, the HIV anti-epitopes on a class 1 molecule. So the first one, I think it seems to be like a very, very tight restriction. The second one, as I said, there are several murine models in which that doesn't seem to be required, the second type of restriction. So we have to be, I know it's always painful, but try to be careful when we talk about class 1 restriction, what we mean exactly with those words.
I also want to add something that make a CD8 T cell based strategy even harder is that putting the transcriptional silence aside, in order to have a T cell to be functional, it requires physical contact, unlike antibodies. And there are a lot of non-lymphoid tissues at the non-inflammatory state are not permissive for T cells. So, and as we know that for HIV patients and are successfully treated with ART, the inflammatory status is really low. So how do we make those T cells get into those tissues is a big challenge. Although we don't have very strong evidence, but I believe naturally you see a lot of reservoirs in the tissue. For example, CNS or genital tract, those are non-permissive tissues for CD8 T cells. How can we push them there? We don't really have a solution. So without being physically at the right place, the CD8 T cells won't make a difference. That's why probably, to my opinion, antibody would be a more promising strategy. Not to mention that the latent virus are carrying a lot of escape mutations to the CTL response. So there are a lot of things to do to improve the T-cell-based strategy.
Other questions? Yes, in the back. And I know it's getting to be, well, it is wrap-up time. So, short answer.
Okay, this might be quick, it might not, but Dr. Shane sort of touched on this a little bit, but just kind of a question for across the panel. What about the brain? So, with Fauci's new initiative, do you think any of these interventions are going to have an effect? I mean, I think it's been pretty definitively shown in the macaques that there is a latent reservoir in the brain. There's a lot of data in there, especially now with the last GIF Foundation that was just presented, that there's HIV DNA in the brain in humans. What do you think what any of these interventions will do for the brain?
Let's start with Janice on this one. And then go walk down the panel.
Well, I mean, I think that the brain is really a very difficult target because you don't want CD8s to go in there because it's going to be really damaging. And I think, as Rebecca said, there's a lot of data now to show that the virus is there in multiple kinds of cells. We study microglia and the perivascular macrophages, but there's also emerging data that the astrocyte really has to be looked at too. So there may be multiple other cells in there too that are going to be pretty hard to, the astrocyte may be very difficult to target because it's infected. It does flare up with inflammation, but it just sits there and is pretty silent without it. So there's a lot of barriers, including the blood-brain barrier to getting things in. So.
Okay. Anyone else on the panel want to address that?
The microglia fraction derived from yolk sac macrophages persists lifelong. And at least from in vitro data, they're highly susceptible to infection. So that means once they're infected, they could be infected for a really long time. And in humanized mouse models, we don't really model that because we don't have the human York sac macrophages. Although under the inflammatory, during inflammatory response, there are small percent conversion from blood monocytes to microglia-like macrophage in the brain, but those are relatively short-lived. So it's hard to study reservoir in the brain in the mice. There are definitely proposals or papers published, but there are always big question marks on those studies or major limitations on those studies. So, and also I think the reservoir in the CNS is really difficult to tackle.
So I was going to add, clearly there are issues in the brain, and the Boston patients and one of the patients that Christine and I took care of rebounded with a meningoencephalitis. Having said that, Timothy Brown remains intact, or his issues don't relate to HIV there. And so I think that we have a lot to learn about which cells in the central nervous system actually get replaced with allogeneic transplant. But clearly for N of 1, it looks possible to eradicate the infection even in the central nervous system.
Yeah, great. It's probably closing time. So let me just summarize what I said at the beginning, and that is the field of research for a cure has really moved by leaps and bounds. And this is all going to be proof of concept that then we can apply to bigger human trials. Hopefully we can get cases like some of the ones we heard, Joel Blankson's case and many others, that we'll be able to get into sustainable remissions. Other cases we may be able to reduce the reservoir size. And maybe with transplants, from what I'm hearing down the end of the table, we may get to a true cure where the virus is no longer present, or at least in limited amounts. The other point I wanted to make is, although this is all cure, we're all talking about cure, all of this science that you heard today does provide a bit of proof of concept for vaccine development, which actually is still way behind. I mean, it focused on CD8s for years, the CTL CD8s. It now is focusing on neutralizing antibodies, and hopefully we'll get a blend of a vaccine. But if we get some home runs from the cure field, might be able to reverse engineer and take that into vaccine trials to prevent HIV. The therapeutic vaccines didn't really work, and Fauci presented, and I think one of you, I think Debbie, you also presented that. But I think we've made a lot of progress. I'm going to remain glass half full. And with that, I want to thank all the speakers from today and the panelists.